Let me take you through a day in my life as the research intern. I start walking towards the plot right when I arrive at the New York Botanical Garden, and we start plotting out what soil we're sampling. So here's me recording the data, and we start sampling. And throughout this time, I'm also the data recorder. And once we get everything, we collect the samples right here, as you can see, and immediately we take it back to the lab and start taking them out. So these are the bulk density samples I'm taking out, and we're basically measuring the volume of the core that we sample from the site. And I am in charge of organizing them and labeling them into the correct bags so that we can measure the mass on a lab day, because after this is the completion of a field day. We also sample composition samples, but the idea of organizing and labeling them is the same, so I didn't show it. So now we'll move on to a lab day. So for lab days, we would be measuring the mass of the bulk density samples and also the composition samples. And we would use sieves to sieve out the bits and pieces, whatever remains on top will go into the composition samples, and whatever gets sieved underneath, like all the soil particles, that will go into bulk density, so that we know how much soil is in each sample and what is the composition of the things that don't end up in the soil. Like, are they roots? Are they stems? We also dry out the samples in order to gain accuracy, and after everything is dried out, we package it into these small conicals. And as you can see here, I am labeling the conical with the plot ID of each sample and pouring it in so that we gain like a sample in the conicals. These are super interesting to package because you can see the different textures of the soil at each plot. Like some, after they are dried, have bigger soil particles, and others have smaller soil particles. Some are like completely just dust. If you want to introduce yourself.
Yeah, sure.
Yeah.
So, my name is Brad Oberle, and I am an Associate Curator here at the New York Botanical Garden. So I am really passionate about plants. I have been for a long time. So my whole research career has really focused on plants, their variety. So I've done some work on plant evolution, a lot of work on plant ecology. So that is, how plants got to be the way they are and how they grow in the environments where they grow today. Yeah. And that is also the kind of work that I do here at the Botanical Garden.
That's cool. Yeah. Okay, so my first question for you is, what inspired you to conduct this soil composition research project in the Thain Family Forest?
Great question, Gina. It was really about recognizing how unique this space is and how much we could learn from studying the soils there, because soils are really important all around the planet. Yeah. Urban forests do a lot besides store soil carbon. They cool our cities, they give us a beautiful place to relax and spend time, and those services they provide are extremely important. So what's unique about this space? The Thain Family Forest here at the New York Botanical Garden is the largest old-growth forest in New York City and one of the oldest research forests in the world. So it has a really unique history compared to other kinds of urban forests. Also, people have been studying it for a really long time. So we've got some great historic data about what the soils looked like in the past. So you put all those pieces together, and that's really what inspired me and the team of other researchers here at the garden, including you, to start looking down at the soils for what they can do for us.
Yeah, definitely. I definitely agree with your points about how the soil is like a reflection of the atmosphere, like the carbon in the atmosphere. It's also interesting that because the Thain Family Forest is in New York and it's an urban forest, I feel like it will reflect the living conditions in New York. So
that is a really great point. And if I can follow up on that, Fiona, that speaks to another part of this project, which is one that I know you're really excited about. It's how the forest does reflect those urban conditions. So all forests absorb carbon dioxide, all forests have soils, but not all forests are in the middle of New York City. And there's a lot about being in the middle of New York City that makes this forest different. Part of it is that the forest is more prominent, but a lot of it too is that urban areas have urban problems. And some of those urban problems include pollution. So some of the really important research in the Thain Family Forest from over 30 years ago showed that there are higher concentrations of heavy metals like lead in our forest than there are in forests in more rural areas. And the things that influence lead in the soil might connect to the things that influence carbon in the soil. So that urban element of this forest is really important too. It complements the basic forest ecology research that we're doing about how carbon moves through the forest.
Yeah. Next question for you is, when did you discover your passion for research and plants?
Well, my passion for plants goes back pretty far. My mom tells me when I was a baby, she used to take me in my car seat and set it under a tree. And I would just look up and watch the tree and be perfectly happy for a long time. So it feels like it's kind of in my DNA. I don't really remember what that was like. What I do remember was being 12 and going for a hike. And it was my favorite time of year, around my birthday in April. And where I grew up in Missouri, April is really a great time to be out in the woods because all the spring wildflowers are coming up. And on this hike, I just started counting the different kinds of plants that I saw and ran out of fingers, got up to about 12 different species. And I had no idea what they were, but the diversity was really exciting. And so I went to a bookstore the next week and bought my very first guide to wildflowers. So that kind of started me on the research career, but that isn't really research experience. The first research experience that I got was when I was an undergraduate student at Kenyon College in Ohio. I had a lot of different campus jobs, and I had an opportunity with my introductory biology lab professor to help him as a research assistant. And actually, he was doing work on carbon cycling in wetlands. So I got to use a really amazing piece of equipment to measure carbon dioxide emissions from soils in different parts of our campus landscape. And it's funny because my research path went very far away from that topic, but has since come back. So it all connects.
Yeah, because that was about carbon cycling, and the research projects, one of the research projects you're doing now is also carbon cycling. So my third question for you is, how do you think your research projects will benefit the community of the Bronx?
The Bronx is a really cool place, and it is a community that really depends on our trees. If you look at all the boroughs in New York City, the Bronx is actually the greenest borough. And a big part of the reason why the Bronx is so green is because of the New York Botanical Garden. But not everybody gets to benefit from the forest where we're doing our work. By doing this work on the forest, my hope is to understand it better so that we can improve the health of urban forests around the Bronx and the really great things that they provide. But that's just one general thing that can benefit our community. A very specific thing that will benefit our community is actually studying what's happening with heavy metals in our soils. So, as I mentioned before, research in the forest 30 years ago found that we had relatively high concentrations of heavy metals. Doing this project, we have an opportunity to see how those concentrations have changed over 30 years. We might find that all the lead that was there in 1989 is still there today. That might be a bit of a problem for our soils, but it's not a problem for anybody else in the community. What I expect to find is that there's less heavy metals in our soils today than there was in the past. So that's probably good for the health of the forest, but it raises a question: where did all that lead go? And the most likely answer to that question is that it moved into our watershed. So the New York Botanical Garden not only has the Thain Family Forest right in the middle, but right in the middle of the Thain Family Forest is the Bronx River. The Bronx River is the largest open waterway that remains in New York City, and it's a watershed that really connects the Bronx and our surrounding communities. So by learning what's changed with lead in our soils in the Thain Family Forest, we can discover whether or not that legacy pollution has been redistributed around the landscape. People in our community deserve to know. and that can help us manage our natural resources better. Yeah, so there's a lot of great basic science there, but a lot of it has very concrete applications.
Yeah.
This is about improving people's quality of life, improving their health, and that is really exciting. It's really meaningful.
Yeah, definitely. Especially how the environmental health, or like the health of the Thain Family Forest, plays a role into the health of the community, which is something I find really interesting.
Well put.
So my last question for you is, what is something that is the most surprising thing you have found from the research so far?
Well, so far we've kind of just scratched the surface. We've collected a lot of soil samples. We started processing them in the lab, but we don't know a lot about our primary research questions. But just from what we've seen at the surface, there are some things that I find pretty surprising. Our soils are really variable. So this is probably something you've observed too, because at every single point we sample, we pull out a soil core. Sometimes in those soil cores, you find a thick layer of organic matter right at the top. Other times there's almost none. Sometimes you can't see the difference. And that wasn't something I'm used to. A lot of the soils where I've worked are soils from Florida, and Florida soils look very homogeneous. So it's not all that surprising to find a difference between the soils that I'm familiar with from the work I did in Florida and the soils that are here in New York, because obviously Florida and New York are pretty different places with a lot of other things that vary between them. But that variation from place to place just within a small forest, I found pretty surprising. And I think it makes me even more interested in the answers to our major research questions. Because I think even within the forest, we'll find places that vary a lot in terms of how much carbon they have, a lot in terms of what kinds of heavy metals are present. And by understanding why they vary so much, I think we'll have some powerful further research questions about what is actually changing across this forest landscape.
Yeah, I definitely agree with your point about like the variability, because like sorting through the soil, like some plots would have like a lot of stones, and then other plots there's like basically none, but like a lot of duff and stuff like that. And it's just really interesting to see like in such a small area there's so much variability, as you mentioned. But thank you so much for being an interviewee today.
It's my pleasure. It was a really fun conversation that I had with you, and thank you for being part of our research team. You've made a really huge impact.
Thank you. I really enjoyed like field work and lab work, especially field work, because I've never really had that much experience in that. So gaining experience from this research project definitely is something that's useful in my toolkit.
Awesome.